All right, so let me just start the live stream. Right. You come here. How are you? Right, guys, so let's settle down. I want to start the paper. Is there a mock exam? No? Did someone stole something? I think you can check the toilets. Right, guys, can you please settle down? Right, so we want to start with the revision of this paper. Is there anyone who still doesn't have a copy? Yes. Is there anyone else who doesn't have an extra, who doesn't have a copy? Right, so let's start. Come again? Right, so guys, so let's start with the paper 2. Can we please just follow it together so that we answer the questions properly and we all understand what's happening here? Right, so the number 1 that we have here, it says figure 1.1 shows a red-billed hornbill, Tropic damarensis, I think, a species of hornbill in the family of your Ucerotidae, something like that. Right, what does it say here? With reference to the scientific naming of the red-billed hornbill, describe the binomial system of naming the species. Yes? Yes? Okay. Is that better? All right. So we want to look at this question. It's asking with reference to the scientific naming of the red-billed hornbill, describe the binomial system of naming species. What is the binomial naming system? How do we name species using the binomial system? Yes. Okay. Yes, the genus, right? And what else? Yes. Yes. So remember, they said with reference. With reference, are we together? So you need to talk about this species, right? So this is the scientific name of that red-billed. Let's see. That's the scientific name of the red-billed hornbill. Are we together? So you need to be talking about with reference from that scientific name. So which one is the genus? Which one is the species? And also how? How it's written. Okay. Right. So let's look at it. Which one is the genus here, guys? With this name, with the scientific name. Yes, right? So remember, you need to explain with the binomial system. You write binomial system. It's written with the genus first. Are we together? And followed by the species. Yes. Yeah. Yeah, it's still fine. Yes, you can take a bit. How are you? Right. Are we together, guys? So the genus. What is the genus? What is the genus? Come again?
The name in capital letters. the first sentence you wrote
No, with reference. With reference. With reference, yes. So you need to tell them which is the genus here, which is the species there. Go together. So what is the answer?
in capital letters.
Right, someone online is saying it's two-part naming system dealing with the genus and species name. The genus being written with a capital letter, right? The red bill, and the species being written with a lowercase letter, one bill. You agree with that? So remember, that is not, Eleni, that is not the scientific name. You have to use the scientific name.
So what did you say? Genus is always in capital letters.
The genus is always in capital letters. Yes, you can explain that. Yes. Yes, you can explain that. But remember, it's only 2 marks, so don't try and waste time writing all of that. Put up? Yes, to underline. So what is the genus, guys? Tropic, right? Your Tropic. And what is the species? What is the species, guys? Can you spell it for me? M-A-R-E-N-S-I-S. Are we together? So we need to show that reference there. Is that correct? Right.
Everything in capital letters. No.
Okay. Underline which part, the whole name, huh? Yes. This is the
Sir.
yes.
Why? No.
Didn't they say capital letters? Guys, one at a time.
Only the first letters of the genus is supposed to be capital.
Only the genus.
Yeah, and the species is not the species.
Oh, okay. I see. Thank you. So it's Trocus.
Sir.
Yes, come again. Yes, online. Belinda.
Sir, can you please scroll up?
All right. Is that okay? Which part do you want to see?
No, sir, I don't understand. Oh, okay, I see. It's okay, sir.
Okay. Is that fine, guys? Is that fine?
Yes, sir.
All right.
No,
So
no,
can,
sir, no, sir. The species is in small letters, everything.
everything.
Yes.
So even this part, the D.
Yes.
Okay, I see. Like that. Right. Wait. It's for the mic.
So how much did you
Yeah.
want?
It's only 2 marks.
No.
How much they cost? I don't know, to be honest. Right, part 2, guys. Describe 2 features which identify the red-billed hornbill as a bird. Yes. The feathers. Right. What else? The legs. What do you call them? Scaly? Scaly? I'm hearing scaly feet. What else?
So the legs are not scaly.
The beak. All together. So any of those, you get your mark.
The wings.
The 4 limbs modified to form wings. Yes. So mentioning the wings as well.
2 pairs of wings.
The wings. 2 pairs or 1 pair. Guys. The wings. Right. So any 2 from there, I think you'll get your mark. Right. Are we done with that question?
Yes, sir.
So can we move on to the next one? Right. Page 3, yes. Are we all there? Right. Now it's just figure 1.2 shows a food web involving a red-billed hornbill. Right. You've got your food web there. So you need to identify your sources, guys. First, identify the producers. Are we together? You've got your leaves, you've got your fruits, you've got your seeds. Yes. Can you bring the pile here? Is it the one? No. Where is it? Check that one. Yes. Should have a picture of a bird. Right, so guys, let's look at that question. So what is it saying here? State what the arrows represent in a food chain. Yes. The flow of energy. It represents the flow of energy. The flow of energy. Are we together? You get your mark there. Right. So please, if they ask you to describe the features of a food web, what do you say? What are the features of a food web? No. What do you, when they ask you to describe features of a food web, what can you say about food web? Food chains are tropic levels. They're asking you to particularly describe this, what's happening here.
So we have many.
Yes, several food chains could be interconnected. Are we together? That's one thing you can say to describe this food web. What else do we have? What else does it make it distinct from food chains? What difference is it? Exactly. So you can have many sources for each tropic level. Does that make sense? Yeah. That's the other differences. So the several interconnected food chains, and what else? Many sources for each tropic or each feeding level. Does that make sense? Those are the key features of food webs. Please take note. Right. The next part, write down the longest food chain from this food web. There are two. Okay, let's try and find the longest one. Let's try to find the longest one. Right, so the guys online, what are they saying? Here's the chat. Let me do this. Right. So here is saying the leaves, grasshopper. So if we start at the leaves here, we go to the grasshopper, then the wig, the red-billed hornbill, then the monkey. So that's 5. Is there anything that has more than 5? Is that, oh, it's 4. 1, 2, 3. Oh yes. Sorry. There's the seed, the grasshopper, the red-billed, then the monkey. That's 1, 2, 3. So they want to take it. How is it called? You said seeds, grasshopper. Oh yes, thank you. 1, 2, 3, 4. Yeah. So I think I'm making a mistake of not counting the producer. Right. So you take your leaves to your grasshopper. Don't forget to show your arrow. Grasshopper. Then where to? The red-billed. Just minimize this. And then what else? Monkey, right? And then what is the other one? There are seeds.
Should be right.
You can write any of them. You can write any of them, you get your marks. So remember, you just need to show that there are 4 and then the arrows, they'll give you your marks. That's where the 2 marks are. Any of those 2. So seeds, grasshopper, then to the red-billed hornbill, to the monkey. Are we together? So any of those 2, you get your marks. Now we copied that down. Have we all copied that down? All right, so can we move on? Now, part 3, explain why there are usually fewer organisms at tropic levels furthest from producers. I would like someone to give me the answer for this one. A well-explained answer. Yes, capire. Guys, can you keep it down? Yes, capire. Yes.
And then also some things must be.
Yeah, you got the point. There's materials that are digested by your organisms. So they're passed out and they're lost as energy. Are we together? Did you all get that? Yes, give another way.
So I feel like the furthest tropic levels produces at the least energy, which like get the least energy. So that's why they are the fewest. Like the one closest get the most energy.
Okay. Right. I'll stick with Capille's answer. Yours is a bit. I'm not too sure. Right. The answer is that there's energy lost at each feeding level altogether. Right. So remember, you now need to mention the ways that energy is lost altogether, which is your undigested material, respiration, movement of the animals. Right. Those are your ways the energy can be lost in each feeding level. That's why there's fewer organisms at each feeding level. Does that make sense? Yes, that's the answer. Energy is lost at each feeding level. At each feeding level. Yes, you can. Tropic level is a feeding level. The highest ones.
Yeah, like mine will get the least energy.
Yes, that's why there's fewer organisms. But then the answer is mainly this. But try and put it in this way. How is this? Do you even know what sex sisters? All right. Right, so can we move on? You're just saying things, huh? Oh, okay. And we copy it down. Can we move on now? Right, so we're now moving to part C. Guys, let's keep it down. Right, what if I said that there's hardly energy to supply the next chain? You need to state why. You need to state why. That one is not really answering. Yeah. Okay, the red-billed hornbill, your T. damarensis, is an insect-eating bird. The beaks of the birds are shaped according to the type of food they eat, right? Figure 1.3 shows different shapes of beaks of some birds. So we've got your A, B, C, and D. Those are your characteristics. So please note, with such questions, you need to look at the physical characteristics. What you can see. Are we together? Now they ask you, use the key to identify the genus of each bird. Write the letter next to the genus name in the table 1.1. Right. Right. So we all know how to use a dichotomous key. So I think we looked at the question also on, was it Thursday? To do with a dichotomous key. So what does it say in here? Big straights, go to 2, and then big curves. So big straights, which ones have straight beaks? C and D. Are you sure? It's A and D. These are the ones. A and D have straights. So we take A and D to 2. Are we together? Now we're trying to distinguish between A and D. Are we together? Now we ask, beak at least 3 centimeters longer than its width, right? It's your pelecanus. So which one has a beak that is 3 times longer? D. Right. So the pelecanus is D. We put our letter there. Are we together? Now, beak less than 3 times longer than its width. It's the other one, which is A. Now we move on to the ones that were curved. Right? Both parts of the beak are curved. Is your cytacus, something like that. It's your, is it B? Both parts of the beak are curved. Can you see the bottom part is also curved? So we put a B. Where is it? This one. And then the only upper part of the beak is curved, which is your C, the remaining one. Are we together?
So we don't know.
Yes, Uri?
So
What's the question? Isn't B supposed to be at C? No, both parts of the beak are curved, is your cytechus. So if you look here, the one that has both parts curved is the B. So that's why we put it at your B here.
oh, I see.
Mhm. Are we together? Online people, are you there?
Yes, sir.
Right. So we quickly name those species, and then we can move on to the next part. Right. Now we look at number 2. Number 2 is saying 6 identical pieces of onion epidermis were displayed in salt solutions of different concentrations. Guys, are we listening? After 5 minutes, they were removed from the solution and observed under the microscope. Figure 2.1 shows diagrams of these pieces of epidermis as seen through the microscope. Right. So here we've got your solution 1 and 2, 3 and 4, and 5 and 6. Right. Okay. So can we see what's happening here? What are we doing to the epidermal cells? What's happening here? Here, water is being lost by the cells, right? So we've got different types of solutions here. So sometimes they can give you one way, it's a concentrated solution with your cells inside it. What type of solution is that? Hypertonic, right? That's a hypertonic solution where your cells are losing water. Are we together? That's a hypertonic solution. And then the one where your cells getting more water, that is hypotonic. Please remember that. Then how about the one where there is no movement? Yes. Yes. As in life. Oh, yes, yes, yes. Life when they burst to open, yes. Right. Now they ask you, what is plasmolysis? What is plasmolysis? Yes. Yes. Yes, the cell loses water. Are we together? Yes. And shrinks. Please note the shrink part. Loses water and shrinks. Nothing. I was laughing at my handwriting. I can't write on these words. Right. Now they ask you, explain why plasmolysis occurs. Yes. Yes, you.
This is when there's a low concentration outside the cell of the So inside the cell,
Yes.
there is a higher concentration.
Yes.
And outside the cell is a low concentration. Meaning that water will now be churned out of the cell
Into the solution. Great. Right. So you need to mention this happens when there is a lower water potential outside the cell than inside the cell. Are we together? So water molecules move out of the cell through osmosis. I'm sorry I changed, but then your answer is close enough. They'll give you the answer. So through osmosis. Are we together? So that is the answer there. What is the guys on chat? Water moves out of the cell. Yes, correct. That was the first question there. Right, so quickly copy that down and then we move on. Sorry? It was JJ. I don't know. Was it JJ? Oh, yeah. Quickly. After lunch. As soon as we finish this. What do you mean? It's just 45 minutes. It's just 45 minutes, guys. Wait, so which one do you want to write the mock-up? Is it the paper one or the paper two? Paper two are going in together. You want to do both of them? Yes, I'm sure. There's one case that's missing for the paper three, so I'll still look at it now. Yeah. You want to write paper two? Paper three. Guys, okay, this is highlighted. Since we're doing paper two, you cover most of the topics. So therefore, paper one, I'm using it to test you guys. Are we together? Does that make sense? Yes. So paper three, we'll look at it together. Right. Can we move on? Right, number the B. Use information in Fig 2.1 to complete table 2.1. Right, so you've got the salt concentration and the various concentrations of those solutions. Then the total number of cells seen in the field of view. Number of cells plasmolized and percentage of cells plasmolized. Oh, yes, so we need to go back to this drawing, huh? So we need to be able to count.
Solution 1 is 20.
It's
100.
I can't hear what you're saying. What are you saying?
20, 0.
Right. So can we start with this field here? Right? Your solution 1. What is the number of cells that you see? 20. Thank you. Why are you saying 20, 0? Oh, okay. Yeah. I'm wondering what's happening. If you're having a seizure or something. Right. What's the solution 2? How many cells do we have? 20. And then here? 0, 0. And then here? 19. Do we give? Wait, solution 3, guys. How many cells do we see?
They are 19.
They are 19.
There's only one that's plasmolized.
So we put 19 there. And then number of cells plasmolized? And then percentage?
No, for 95%.
Number of cells?
I mean, 5%.
5%. You guys are nuts. Right. 4. It's how many? 4. Yes.
16.
The percentage?
20%. 20%.
It's 20%. How is it 20%? 16 out of 20. You're looking at the percentage of cells plasmolized. 80%. Thank you. Guys, your maths needs work. Then the number of cells for solution 5. Number 5. 1. And then the number of cells plasmolized? 19. What is the percentage? And then the last one? Is it 20? 0 here. 20. And then? 100. Please, guys, please don't confuse. Look at what it's asking for you in the table there. Please take notes. Are we together? Right. Now they want us to draw a graph. They want us to draw a graph of percentage of plasmolysis against concentration of salt solution. Right? So which one is my dependent? Which one is my independent? Concentration is where? Yes.
So the first part, the total number of cells only the one. But they're asking for the total number.
The total number of cells seen in the Okay. Yeah. So it will just be 20, 20, 20, 20, 20. Yeah, because they're saying total number of cells seen in the field of view. So it should be 20, 20, 20, 20, 20. Yeah, that makes sense. So it should be 20, 20, and 20. Right, thank you for that. And then everything else is fine. All right.
I don't understand why is it 20?
They say the total number of cells seen. Total number of cells seen in the field of view. So that there, you just look at the cells, you can see this one you even included, that is a cell. It's just plasmolized. If they had asked for the ones that were not plasmolized, then you put your 19 or your 0. Right, so here, your concentration of salt solution is on the x-axis. And then here, the percentage of plasmolized cells is the dependent. Are we together? Since we're controlling the concentration there. So now we want to choose a scale for this. What is my scale? What scale can we use for 0 points? So can you see it's just moving up by 1, by 0.1.
You can make 25.
We can make 25.
Kind of blocks.
Zero point? Yo, what's going on?
Wait.
0.05.
Percentage can be 25.
All right, so here we start from 0, and then here it's 0.5. It won't fit. You can just say 0.1. Let's see, 1, 2, 3, 4, 5. Yeah, 0.1, 0.2, 0.3, 0.4, 0.5. That scale is fine. So you see that it's covering most of the, yeah, all of it. Right, now percentage of plasmolized cells. Let's look at the percentage. We're going from
25.
okay, from 0 to your 100. So here we use 25. So here 25. Here 50, 75, and then 100. Right, so let's plot. So please, before you plot, make sure you know what one box here represents. Are we together? So here we've got 0.1 to 0, meaning 10 boxes here. You divide your 0.1 by 10, it gives you 0.01. So one small box represents 0.01. How about here? Divide 25 by 10, 2.5. So if this part here, each box is 2.5. Right, now let's plot our graph. So we want to start from your 0.0, which is that point there. You can still just put your x. And then the next one, 0.1, also still 0. So we put your x there, right? Sorry? Yeah, you can. Come again?
So if they don't tell us what that is,
Just hold on. We'll see what they want us to do before the question. They just asked us to plot. We want to see what happens. So here, what is the percentage? 5, right? So it's going to be there, right? Do you agree?
It's the second block.
Yes.
Yeah.
2.5 plus 2.5. Yeah, you can make sure. 0.3. 80, right? So we go all the way up here. This is your 7.5.
The second block after 7.5.
Correct. So there. And then 0.4, what do we have? 95, right?
Yeah.
So 0.4, 95, this is 80. That's going to be
2 blocks under 100.
2 blocks under 100. Yeah, true. And then your 100 is the last one, so just, okay. Can you see that? So that is the line there. Are we together? So now they ask you, so with this one,
Mr. Simba, just a question here. I want to quickly duplicate the paper one there. How many learners do you have there with you?
let me count.
Were those copies enough, the ones I made, or the leftovers?
Yes, they were left over.
Okay, how many leftovers? Because I think the copies were about 27, 20, yeah, somewhere there, 27, 29.
You're 22 here.
Yeah, 22, okay.
Yes,
Good. I'll do that then. Thank you so much.
All right. Okay, so this one, I think we can just connect them with using your free hands. Are we together? I think when you get this part here, I think drawing a straight line is fine. Using your ruler. Let me see. The curve is better. It's almost like your cumulative frequency graph, huh? Like that, huh? So also make sure it touches every point. They don't want to see this. They don't want to see that there. So make sure it goes through the points. Right, now they ask you use your graph to determine the concentration of salt solution, which gives 50% plasmolysis. Yep. So you go to exactly, touch the line, then come down. Yes, you have to show those lines on the graph. 0.25, huh?
Yeah.
Did we all get 0.25? What did you get? 0.25, 0.26, huh? So yeah, that's that range. So if you say 0.25 plus or minus your 0.0, 2.24. Okay, so I think you can say plus or minus 0.02. Are we together? Plus or minus 0.02. That would be the range for that. Right. Let's move on quickly. Have we finished this drawing of the graph? Do you understand what's happening here? All right. So if we all understand, we move on. Who's not done? Oh. All right. So please take notes. Whenever they ask you, guys, can you please pay attention? This, yeah, when they say against, you know that this is going to your x-axis. Are we together? And then this is your y-axis. Because if you swap that around, you'll get different answers. Are we together? But then I think you might be able to get correct answers, but then they will mark you wrong because of your switching of your axis. Are we together?
Yes, sir.
Right. Are we done? Are there people still drawing? Now with gifts, huh? Kapira, why are you fighting with everyone? This is not here. All right, the guys online, are you guys fine with the graph?
Yes, sir.
Okay, so please make sure you're also able to do it either on your phone, your laptop, or your iPad, whatever you have. Try to draw the graph as well so that you follow with us. All right, so can we move on now? Now, number 3, name the basic units required for the synthesis of the following large biological molecules. What would you need to make these large biological molecules? Your 3? I think you can just say fatty acid and glycerol, huh? Yeah. That's your glycerol. Right. But then here, if you did your organic chemistry, you know these are your carboxylic acids, this is your alcohols, right?
Please,
Now,
we're done with them.
You're not done with Kim. Guys, it's an interconnected. Don't crucify me. Let's move on. Glycogen. Glycogen. Come again? Guys, remember, yes, thank you. No, I was shocked with what you were saying. You said carbohydrates. Right, glucose. Remember when you're trying to take your blood glucose levels down, right? It is you absorb it into the liver and muscle cells, and then join to form your glycogen. Are we together? That is your insoluble form of the sugar there. Right. Now proteins. What do you join? Thank you. Amino acids. Yes, JJ, we know you're there. Right. Figure 3.1 shows the label from a biological washing powder. Right? We've got contains protease, amylase, and your lipase, your enzymes, more effective than ordinary detergent, not suitable for leather and silk clothes, avoid using boiling water or use warm water, removes blood, egg, and chocolate stains. Now they ask you, explain why the biological washing powders are more effective at removing stains made by blood, egg, and chocolate compared to non-biological washing powders. Yes.
Because they contain things that
Yes.
they bring down.
Then, come again.
Because we enzymes which break down the insoluble blood molecule into soluble one.
Yes. So remember, you need to specify what's in blood, what's in egg, what's in chocolate, right? So you need to state that they contain your proteases. This is a carbohydrate. This is a lipase. So your eggs, right? Eggs have proteins, fats as well, right? Right? Blood has what? Protein. That's protein. Are we together? And then what's the other one? Chocolate has? Sugars. Right. So now you mentioned that your biological washing powders contain the enzymes required to break these down. Enzymes break these down. Come again? What biological washing powders contain enzymes to break these down? Right, so you can even, without even mentioning this, you can say your biological washing powders contain your carbohydrates, your proteases, your lipases to break down the blood, which contains proteins, egg, which contains proteins, and chocolate, which contains sugars. Guys, I'm just giving you ways you can answer, right? This is the way I would have answered, right? But then there's another way of saying your biological washing powders contain carbohydrates, proteases, and lipases. They will break down the protein in your blood, the proteins in the sugar, and the sugars in the chocolates. That's another way of answering. Does that make sense?
So can you, after you mention those stuff, can you just say the enzymes break down?
Yes, you can. You can. This is 2 marks. Is it too much?
So
Is too much?
So what Roscoe said is wrong.
Which one?
Roscoe said.
What did Roscoe say? Yeah, it's a good question.
Too much like that.
Yes, it's too much. What?
Okay.
You're saying it's too short?
This one.
Yeah, this is what I'm saying. This is how I would answer. Because I want to explain that the eggs have the proteins, your blood has the proteins, your chocolate has the sugars, and then the enzymes will break down these things. Yes. Yeah.
try the following.
It's still correct. It's still correct. Right, now they ask you at which temperature should the water be for the biological washing powder to be most effective? Yes.
Definitely below 100.
You won't get a mark for that.
Should be exact.
40. You won't get a mark.
19.
19.
19.
The water has to be warm.
37, right? Body temperature, guys. Body.
So you know what you think?
Guys, remember, for what? For the enzyme. But then here, when we're dealing with your proteins, yes, but breaking down what's in the body all together. So these enzymes are the ones that are used in our body. So we use body temperature all together.
So, sir, do we just like body temperature to realize that?
So what is my, sorry to computer, what is my body temperature?
36 degrees. 37.
37. Do you understand why it's 37 degrees Celsius? Okay, let me explain. Guys, these reactions that take place when we're breaking down the proteins and when you digest, right? Where is it happening? In our body, right? Yes. So that's why we use body temperature for the same type of enzymes. The same type of enzymes. Because they work best at those temperatures. Does that make sense? That is the optimum temperature.
So, sir, in the example, like body, optimum body temperature.
Yes. Optimum body temperature. Guys, what's Nigeria? Body temperature is 37 degrees Celsius. Yes, that's what I specified. No, you have to specify. You have to specify. Are we together? The guys online, are you guys fine? Are you guys okay? Please take note. That's why we use body temperature at 37 degrees Celsius. These reactions happen at body temperature because it's in our body, right? The digestion happens in our body. And remember, you guys, if you have a fever, what happens to you? You become, you're not able to move, right? Because you're not getting enough energy. That's why they use that towel, they put a towel on you so that you reduce your temperature. Does that make sense? The towel that they put on you when you have a fever. You want to put the towel on you. There's no other case for you. Huh? Explain now. Can we move on to the next question? Are we now fine here? Okay. Explain why the manufacturers of this biological washing powder advise against boiling water. Yes. Yes.
So what happens?
So it's more effective.
Yes.
So if you use boiling water to destroy the active substance, the active side, the active side, then it means it becomes natural.
The substrate one.
Right? So what happens when it becomes denatured? What happens when it becomes denatured? It will not. What will not? What's ESC? Enzyme substrate complex.
Hey.
Right.
Little question.
Also, JJ, I think your mic is on.
Oh, sorry, sir.
the or sugars present in the blood, pigs, or chocolate. Are we together? Right, so that's how I answer that one day. Get your 3 marks. So mention that your boiling water is above 40 degrees Celsius. Your enzyme will be denatured. Are we together? When that happens, the active site is altered and the enzymes cannot break down the proteins, the fats, or the sugars inside the blood, the eggs, or chocolate. Are we together?
Right?
Oh, yes. The enzymes cannot, thank you, cannot break down. Right. Are we together?
Yes, sir.
Right. Are you still copying?
Yes, sir.
Please copy down quickly. You hate color, JJ. You hate color the questions. I don't want to be involved. I do not want to be involved. Remember, this is being recorded on YouTube. Right. Yeah, I know, JJ. But then once you get the trick of answering them, you will like them. Are we done copying, guys?
Yes, sir.
Quickly, guys. Quickly. Agnes, can you please stop writing? Where's my eraser? Oh, I know things, yeah. Thank you. Right. Agnes, really? Why are you doing that?
Because that anything right?
Really?
Yeah, is
Is that true, guys? Okay. All right, so Agnes will take it as a mistake, huh? All right, number 4. Can we move on, guys? The cholera outbreak reported in some parts of Namibia in recent years appears to have resulted from using water contaminated with sewage for household use. Name the pathogen that uses cholera. Vibrio. Guys, you guys are making too much noise. Right, so please take note of the spelling of that parasite. Please take note of the spelling. Vibrio cholerae. If you just write, where's JJ? If you just write Vibrio cholerae, you get it wrong. You will get it wrong. It's Vibrio. Vibrio cholerae. Right. Now, what are the state 2 symptoms of cholera? Diarrhea. No, it's not diarrhea. Guys, okay, for me, what I noticed is that it has only 2 R's. Those are the only 2 letters there. Don't completely say there's 2 A's or whatever. It's dia, right? Dia, and then double R and Ria. Ria. This double O. Which double O? This is an A. Are you saying that one? Are you sure? Please double check. Please double check. This
No,
is that. Mamse? Guys, one at a time. Yes. It's an O. It's an O. Diarrhea. Are we together? Yes. What's the next one?
Vomiting.
Yes. Drowsiness. Come again? Losing? No. All right. Drowsy. Or tiredness. Tiredness. Any other ones? Yes. They won't give you one. They won't. Hold on. Yes. Wait. Drowsy. This is a W. This is a W. Drowsiness. Yes. Sweaty. Really? I don't remember. Anything else besides those? Right. Yes, Eleni, we can move on. Name the part of the digestive system which is affected by cholera.
The small intestine.
Yes, correct. Small intestine. Right. And then you can be even more specific. It'll be the ilia. Correct. Now, D, describe how the organism that causes cholera produces the symptoms in the human body. How does the organism that causes cholera produce symptoms in the human body? Guys. Too many people talking at one time. Yes, stop here. It's fine. It's fine.
I think
Yes.
the bacteria was basically now therefore causing the wound.
Yes. Yes. It is.
And then due to that, then that means that your body uses salt and minerals. Therefore, then resulting to your body now retrieving more fat than some other parts of the body.
All right. Yeah, that's correct. So now, guys, there's one step that you missed. Is that the toxin that it releases? Yes. So this Vibrio cholerae releases a toxin into the walls of the small intestines. Cholerae releases a toxin into the small intestines. So this was easy paper, you guys are saying. Right? So what happens here? Once the toxin enters the walls of the small intestine, what happens? What happens? Wow, does the water potential happen? Yes.
Sir, JJ answered in the chat box.
He answered in the chat box. The toxin causes the body to secrete enormous amounts. No, it's this part, the salts. It's the salts. The salts are the ones that are supposed to move back into the stomach, not the stomach, the lumen, and then the water flows out through osmosis. Are we together? So the toxin is the one that causes that, the outflow of your salt. Are we together? All right. So the next point here would be the lumen. The inside, like, yeah. Right. So the toxin causes outflow of your salts. All together into the lumen.
The toxin causes.
Yes, causes outflow of your salts into the lumen, lowering the oh. Thank you. Outflow of your salts into the lumen, lowering the water potential. What's the symbol for water potential, by the way? The trigon. Right?
Jason
Oh, what?
Momoa's sword.
Huh? What was that?
No, the symbol for water potential is Jason Momoa's thingy.
Oh, yes.
Yeah. Well, Aquaman, guys.
Aquaman, yes. The trigon. Huh? No. It's Poseidon, yes. God of the sea. Right. The toxin causes epithelial cells of the small intestine to secrete chloride ions, which diffuse into the lumen of the small intestine, lowering the water potential, causing your water to move back into the.
So is that another point?
No, it's just one point.
No, why you still put the arrow?
Guys, should I show you? This is like how I answer my answers. This is how I show my steps. To show you like what you need to include. Are we together? Into the small intestine. Are we together? Right, so quickly copy that down and then we move on to the next part.
I was just finishing this last two questions.
What are we done with?
No, the thing is almost break.
What time does I don't remember mentioning what time the break was.
12:30.
Okay, so it's still 11 past 12. Yeah, so what's the rush? Your food won't go anywhere. The presence of chloride ion results in an increased amount of water to be released from the epithelial cells of the small intestines. Yes, that is correct. Are we done, guys? Quickly. I want to be able to finish this paper too, at least cover most of it, so that when we come back, we just look at one question and then you write your paper one. Are we together?
Multiple choice.
Yes, multiple choice. There's a worksheet that I shared with you guys. I don't know how many of you did it. The variation. Do you still remember it? You couldn't do it. We can still cover that. We can still look at that paper before you write your exam. Yeah. So it has the variation in the biochemical cycle. It had what else? What was it else? Your human influences on the ecosystem. Purifying water before usage, and okay, yeah, that's the next question. Right, what does it say? Suggest 2 ways by which the transmission of cholera can be prevented. How? Yes. Guys, the back there. You need to computer, come again. What was the answer? To purify. Now I want to answer, kids. Purify the water. How do you purify it? Yes. By boiling or using chemicals. Chemicals. The chlorination to kill parasites.
So you can also wash your
Yes, you can. Washing of hands. Washing of hands before eating or after using toilet. After using the toilet. Are we together? Right. So I know in the age of your pandemic, COVID, most of us are very used to washing our hands, right? I hope so. Right. Right. Now, one of the complications which can result from having the disease, cholera, is kidney failure. People with kidney failure can be given a kidney transplant or they can be given dialysis treatments. Outline the disadvantages of kidney transplants as compared with dialysis. Rejection. Okay, let's quickly write that. Rejection. Yes, you can say tissue rejection. It's the same thing, right? Rejection. What? How does that happen? The rejection. Yes. Yes, so your immune system will recognize that kidney as a foreign body. Are we together? Then it will attack it and destroy it. That's how it gets. Yes.
So,
Okay, what's the disadvantage? What's the disadvantage?
So it's also time consuming.
Come again? It's time consuming?
Because we have to wait for a certain type of.
Yeah. Oh, yes. But then I think all that comes under rejection. You can put that word under rejection. I don't know what. I don't understand how that is a disadvantage. The first thing you need to 10 to 15 years, right? If you have a kidney transplant. Compared to treatment, would it for how long? For longer. Guys, the person with the dialysis treatment, do they live longer than the person with the kidney transplant? They don't. They don't. Yes. Like the rejection part, because they're waiting for a perfect person who fits the body.
No, but in terms of now waiting, process of now waiting for it.
Okay.
So time consuming.
Yeah, exactly. That's my point. Compared, because they're saying compared with dialysis. Oh. Because that person has to wait for it to be clean. Are we together? Yes. Are we there? Is it isolation? Isolation. Okay, yeah. So you can say isolation. What else? Yes, Eleni, I saw your hand was up. What did you want to say?
so when you're answering a question like this, are you supposed to list and then explain, or just list the 3 disadvantages and compare them with, like, for example, rejection, but with dialysis, there's no chance of that because it's a machine? Or am I supposed to be like, my question was asked, should I list and explain or just list the 3 disadvantages?
Yes, you can first list them and then you can explain how it is a disadvantage compared to dialysis. So here, rejection, the fact that you can lose the kidney instead of dialysis where you can what? They just an exchange. There's no chance of that happening, like you said. Isolation, yes.
So, like, it's not like fully changed. Like, there's not a whole thing that you can survive after the
I didn't get that.
I mean, so like, in the kidney transplant, it's not always where you can survive. There's a chance that you can also die.
Okay, yeah, that's the rejection. Let's go under rejection. Is it expensive? What's expensive? Dialysis is more expensive. Yes.
Like after the way I answered
Yes.
was that I first like came or like first compared the kidney that is better to kidney and then compared to dialysis.
Okay. So you said with your kidney, you mentioned.
Yeah, we think compared to dialysis.
Okay, yes. All right, so here with the rejection, what would you say? With rejection, we say there's a chance of the tissue being rejected. Which can be fatal, right? Which can be fatal. Which can be fatal. Was compared, unlike with with dialysis. What's happening with the screen? It's better now. I don't know why this brightness was reducing. All right, so guys, what was the next one? What was the next one?
Sir, why is that thing so long, man? It's just 3 months.
Wait, we're outlining. We're outlining. Yes. Uri, you don't like to write, huh?
You want to get to the point.
Yes, we're getting to the point.
Like that is an introduction.
How's that introduction? I'm just explaining why rejection is a disadvantage to dialysis. That's all. The dialysis where there is no. Yes, I'm still writing. Yes, I'm still writing, guys.
What's the use of also explaining isolation?
What's the use?
Yeah, we write the isolation. We get them up for rejection, and the explanation.
Yeah, you get them up.
You just write isolation without the explanation. It's still
Yeah, I guess so. Yeah. You still get them up. You still get them up. So isolation, what did we say? What was the answer for isolation? It's what?
Which makes people more sick.
Isn't this for dialysis? Shh, guys. Isn't this dialysis?
Yeah, the machine causes.
Yes, yeah, it's for the dialysis.
You feel numb.
What was the point on isolation? What was it? What's the name? Tashia? Isolation, when you have to, is the point where you're saying you have to wait. Is that the isolation you mean? What is isolation?
So when you
When you feel sick, you don't want people to be around.
So they are vulnerable.
But then that point is only, it seems more applicable for the dialysis more than the kidney transplant. Are we together? What other point can we mention, guys, for disadvantage of your kidney transplant? It's finished. Okay.
Sir, it's expensive.
Guys, let me tell you. There's another disadvantage for kidney transplant. Yes. Lala, you listen or not?
Yes.
Pain. What? Oh? You'll be in pain. Not too sure about that one. That's mentioned under rejection. Yes.
Sir,
Come again, Moses. Hold on. More vulnerable. I don't think so.
sir,
One line. Was it in any?
sir.
Hold on, guys. Yes.
It's no
So what
secret.
about the risk of dying on the operation table like bleeding, infections, and stuff like that.
That still falls under this, guys.
Sir, the textbook says they are more vulnerable.
They are more vulnerable to disease. What type of diseases? Infections. Oh yes, when you mention infections, yes, that's correct. Right? Your more chances. Of infections. Are we together? Yes. Then I'll take it. Guys, you can't make it too much stress. Yes. To isolate. I don't think so. Isolation, you said that the person has to stay away from people who are sick or from normal people. Right? So that causes depression. So please. Surgical complications. It's also part of the injection. Yeah, you can put it under the infections one as well. But then I get your isolation point. Like the fact that you're coming from a surgeon, it has to be kept clean. Are we together? So that there's no chance of that infection growing on that kidney. Are we together? Yeah. Anything else? Okay, so quickly copy down.
So it's still like
The doctor might not like experience my
Yes, yes, Nusiku. Yes, thank you. Yes, that is correct. It is also part of the surgical complication. Which was your answer?
So why are we making it a third point, sir, if it's already part of the fatal?
No, it's actually not similar. This one is tissue rejection, right? Your kidney is not compatible in your body, but then here there's a chance of it being infected. Since it was surgery, they opened your body, there's a chance of your bacteria entering your body through that wound. Does that make sense?
Yes, sir.
Yes? Anything else? Can we move on? Are we okay? Right. All right, thank you. Figure 5.1 shows the cross section of a human eye. Use lines labeled E, F, G to show the following structures. E, the structure that controls the amount of light entering the eye. Right, so we look at that. Which one controls the amount of light? Is it? Yeah. Where is it? The first one, huh? Did you say pupil? The iris, thank you. The muscles here. The pupil is the center part. It's the size of the hole. So the one that controls is these ones. The iris, right? Which is your E. Like this. Are we together? I think so. You have to label both. Because both of them are being used to control. No, this is explaining to name to show you what it's called. So E, yes, you can put your E, and then me, I'm telling you that this is the iris. So even if you leave with E, you get your answer. Yes, E is the correct one. You mustn't what?
Like, I don't know what
You mustn't join the line. Really?
Yeah, it's not allowed.
Guys, yes, sorry, Luis. Yes. Really? I didn't know that. Okay, so we draw with one line. Okay. So you just draw one line to your E. Okay. And then the next part, guys, let's quickly move on. E, if the structure that changes the shape of the lens to focus on objects. It's which one? The structure that changes the shape of the lens to focus on the sun. Yes. This What are they called? Suspended ligaments. Suspensory ligaments. Uh-huh. They don't what? Oh, yes, yes. The structure that changes. So these ones are holding in place. Which ones are this? This one, right? Ah, yes. Thank you, thank you. So it has to be this part here. This part. So make sure you have that part. It's that. Both sides.
One side.
Okay. All your labels should be there. So like this. Okay. G, the structure that refracts the light rays. Which one is the second part? Yeah, I remember. So this one. So what is the answer? G. Yeah. Yes. Come again? We were saying that. Where is that? They're just putting F and G. This one is your. All right. This is the. You said it's the ciliary muscle. And then your G is the. What is it called? No.
G is the lens.
G is the lens. This, the one that refracts. Didn't they say refracts the light? Refracts the light rays.
But that is the lens. That one, the G.
The lens focuses on light on the retina and the cornea refracts light.
The cornea refracts light. So this is the cornea.
No. This one.
So this is wrong. The lens is not what's bending the light.
Yeah.
Really? I thought it was the lens that actually bends the light.
So the lens helps the eye to focus on objects.
On focus on the objects. And how is it doing that? Doesn't actually reflect the light.
Yeah, but
It focuses light onto the retina.
Onto the.
Retina.
Okay, so where's the cornea?
cornea is here, the second line. Yeah. No.
Which one? Here. So this.
Yeah, the cornea helps to refract light rays.
Oh, this one here, in between.
Yeah,
Okay. This one here.
Yeah.
Okay. So this is G. Cornea.
And then we are.
There's a lot going on here.
All right. Let's finish this question, then we go. What's the remaining? Yeah. Let's finish this question and then you can go. Figure 5.2 shows the changes that occur in the eye in response to changes in light intensity, but in bright light and in dim light. Describe what happens to the pupil size and explain how the muscles of the iris cause this change in bright light. Right. So they want you what happens from your bright lights?
So in your bright light, the iris becomes fuller.
So the circular muscles of the
Yeah.
iris contract and the radial muscles of the iris relax. Then the iris the iris becomes larger and the pupils
The
constrict.
eye becomes, in bright light, the pupil becomes smaller. Are we together?
Yes, sir.
Becomes smaller. And then we need to explain how this happens, right?
The circular muscles of the
Circular
iris contract.
muscles
Of the iris contract.
act
and the radial muscles relax.
Radial muscles relax. All together.
And the iris becomes larger and the pupils constrict.
The iris becomes larger.
And the pupil constricts.
And the pupil constricts. Okay, so that's how you answer it today. So remember, when you have your eye, they have those circular muscles like this, and then those muscles going radial like this. So these ones in line, these are the radial muscles, right? Radial muscles, and then the circular, these are the circular muscles. Right. So what happens is this: when they want to make this smaller, let's say this black part smaller, right? It means that these, the longer ones that are holding that pupil in place, they have to relax in order for it to reduce in size, right? And then the circular ones, these are the ones that contract. And then when they have to get bigger, these circular muscles now are the ones relaxing, and then these are the ones pulling it apart. Are we together? Mm-hmm. Now name the type of photoreceptors that enable humans to see. All right. So I think you guys, we can take a break, and then we'll try and finish the paper when you come back. While when we're done, we can then look at that paper 1. I don't know, with the guys who are at home, when do you guys want to write your paper 1?
Write it.
Yes,
So can I write it tomorrow?
You want to just then,
So because I'm coming tomorrow. So.
so you can let me know when you guys are able to write the answer scripts with you. Go through it by yourself, or you can come here tomorrow and write it, and then we can look at the answers together. However you prefer it. Now it's break time. You guys can take an hour's break for your lunch, and then we'll come back later on to cover the rest of the paper. All together. Just 1 hour. Yeah. Right, so you guys are free to go. See you later.
Sir? Sir? Hello? Sir? Sir?
There's no one there anymore, bruh.
But I just wanted to ask, like, should we use the same, like,
Also,
the
what
link?
if
Yes, Nusiku, I can hear you. Sorry, I was muted.
So,
No,
do we
you
use
just-
the same link when we connect again?
Oh, okay, we'll send the links.
No, just the same link, no?
Oh,
Okay,
yes,
sir.
yes.
Yeah, just use the same link. It will still be active.
Okay, sir.
After 1 hour, so by 1:30, no?
Okay.
Yes, sir.
All right, see you.
How was the exam?
Sunday, I don't know what